Okay, in this video we're going to talk about the nucleus. Remember, it's our largest organelle, and it contains the genetic library or blueprints for the synthesis of nearly all of your cellular proteins. And the reason why it's nearly all is that your mitochondria also contain some genes. Now, the nucleus can respond to signals that dictate the kinds of amounts of proteins that need to be synthesized, and most cells are at least uninucleate, or one nucleus. But certain cells like skeletal muscle, bone, and liver cells can be multinucleated, which means they have many nuclei or many different regulation centers. There are some cells like red blood cells that are anucleate, or which means they have no nucleus, because while the red blood cells are developing in bone marrow, the nucleus is actually exocytosed. Now, in terms of the structure of a nucleus, we have three major things. We got an envelope that surrounds it. We got the nucleoli within it, as well as the chromatin that's in between. So just looking at the general structure of a nucleus here, you got the nuclear envelope that surrounds the whole nucleus. It actually serves as a nice protective barrier for the inner contents, which are sensitive. Things like chromatin, which is essentially DNA and packaging proteins, and the nucleolus, which is a place where ribosomal RNA is synthesized. Now, these components are sensitive and could potentially be damaged by some of the other types of enzymes or molecules you find in the cytoplasm. So it's important to compartmentalize then the inner contents of the nucleus from the rest of the cell. Now, the nuclear envelope is this double membrane barrier. It encloses this jelly-like fluid called nucleoplasm, and the outer layer is continuous with rough ER. And the reason for this is that some of the blueprints for protein synthesis travel from the nucleus straight over to rough ER, where proteins can be made. The inner layer of the nuclear envelope is called the nuclear lamina. It's basically a network of proteins that maintains the shape of the nucleus and acts as a scaffolding for DNA. Now, the way that molecules communicate or pass between the nucleoplasm and the cytoplasm is through these nuclear pores, which are basically small channels that allow substances to pass in and out of the nucleus. They're guarded by these nuclear pore complexes, which regulate the transport of specific large molecules. Now, this is just showing a freeze fracture of the nucleus. You can see that it's got two layers here. You got the outer membrane and the inner membrane, and it's studded with these nuclear pores that aid in the transport of material, small materials rather, from the cytoplasm and deeper in the nucleus with the nucleoplasm. And if you look closely, these are what the pores look like. So you can see that they're small little channels with a central kind of aqueous-filled core. Now, looking at the inner nuclear lamina, this is what would be adjacent to the inner nuclear membrane, and it actually supports the structure of the nucleus and makes sure that maintains its shape. And it also serves as a nice scaffolding for DNA to attach to. So nucleoli are these darkly staining spherical bodies within the nucleus that are involved in the synthesis of ribosomal RNA. Remember, ribosomal RNA is essentially what helps to catalyze the reaction of protein synthesis at the level of ribosomes. And it's associated with this nucleolar organizing region that contains DNA and it codes for ribosomal RNA. And there's usually one or two of these nucleoli per cell. Now, chromatin, which is found in the nucleoplasm, consists of about 30% thread-like strands of DNA and a majority of histone proteins here, which actually serve as a nice sort of packaging type of scaffolding for DNA, and some RNA as well. Now, they're arranged in fundamental units called nucleosomes, which are basically where DNA wraps around these histones, and chemical alterations of these histones have an effect on DNA and whether or not those genes can be expressed. Now, chromosomes are what we call chromatin when it's condensed. So when DNA and its packaging proteins are very tightly wound together, they can actually be coiled into a chromosome, which is basically one molecule of DNA. It's an entire strand of a DNA polymer, and when it's in its condensed state, we call it a chromosome, which is visible under a microscope. So in a future slide, we'll see how when the cells are dividing, chromosomes become apparent because the chromatin condenses in order to be able to transport that DNA. So what we're looking at here is basically chromatin and how it coils around. We see that each of these little blue things is a histone protein, and shown in purple is actually going to be the DNA as it's coiled around these histone proteins. Now, What happens is that when DNA is packaged in its histone proteins and tightly wound, or when the chromatin's condensed, it can be condensed in this very tightly wound package called a chromosome, which is what we see here. And this is essentially the visible form of chromatin in its condensed state. During cell division, or when the cells are dormant, they're going to be in this state. If you need to express genes and be able to use them, they can't be in this condensed state. So living and active cells have their chromosomes in their relaxed chromatin state, and they won't appear like this. There's just going to be kind of general haziness within the nucleus. So, an example of what we're seeing here then is where chromatin is, somewhat condensed, not fully condensed, but the fact that you can see some of these darkly staining or kind of darker regions here suggests that the chromatin is condensed. And you can see just the general structure of the nucleus. We got the nuclear envelope, nucleolus, and then chromatin that fills the rest of the nucleoplasm.